we're going to pick up here is we're going to start talking about now the different parts of a neuron and a little bit how things are moving around within these. So if we're looking at this one here, a neuron is going to have a few different parts. It's going to have its main cell body. Again, all this obviously surrounded by a membrane. This is really where that neuron is going to decide whether or not it's going to conduct an action potential or not. We're going to see it's really at the end of the cell bodies that's going to the axon is the determination on a lot of these ones that are going to produce impulses or not if it happens there. So has plasma memory surrounding it, cytoplasm surrounding the nucleus. The area surrounding the nucleus is considered to be the perikaryon, the cytoplasm within that cell body. You can see the cell body, this lit portion right here. We are then going to see we have the specialized start of the axon called the axon hillock. You have the much larger process going on as an axon, and then there's a lot more numerous smaller ones here called dendrites. And we're going to be looking at each of these ones. So the cell body generally is going to be very granular looking in a neuron. This is because there is a lot of endoplasmic reticulum. They were a lot of times refer to this as Nissl bodies, but this is where we're doing all that neurotransmitter, which is usually proteins, so that protein formation. So this is really rough endoplasmic reticulum in a lot of this area, but it becomes visible under the microscope, these Nissl bodies. You also see a lot of times a very prominent nucleolus in the nucleus because, again, these cells are doing a lot of protein synthesis, therefore they need a lot of ribosomes. So the nucleolus is part of building a ribosome. So again, that a lot of times is a little bit more prominent on this as well. So these endoplasmic reticulums are making a lot of the neurotransmitters, which are going to be important for sending signals from one cell to the next. So you can see the cell body or soma is this main area right here. This is what we call a multipolar neuron. We're going to see there is different types of neurons, but this is the most common type. Generally, there's more dendrites than neurons, so a lot more inputs. The axon is more the output, but these dendrites, if you do dendros in Greek, it means tree. So as you kind of look at these dendrites, they look like a lot of tree branches. There can be, like I said, in most cases, there's multiple ones of these. There is certain types where there's only one coming in, or in certain cases where it's really not dendrites coming off the cell body. We'll see there's some different types. But this is one of the ways that processing happens. We get a lot of surface area with all this branching. You can have synapses with a number of different nerves. These can all act as inputs. And again, with the axon, there's only one main output. So processing is taking multiple incoming information and determining one signal to send. The axon is that outgoing process, generally larger, bigger diameter than the dendrites. It has a kind of triangular portion where it narrows down called the axon hillock. This stuff is going to be important to us in a little bit when we start talking about nerve impulse transmissions. But this is what's going to go and connect with other neurons or muscles or glands. They may branch at the end into these collaterals and multiple branches at the end. But again, you can see there's only one main axon leaving. It does not have Nissl bodies in it. We call it the axoplasm in there. And we call the membrane the axolemma versus the plasma membrane. Again, it can branch out a lot at the end to these different collaterals, and then you a lot of times are going to see these large kind of extended ends on there called axon terminals or telodendria. These synaptic knobs are going to be where we interact with another neuron, usually, like I said, a little bit expanded there, and it's going to be this area where there's going to be a number of vesicles in there that when its action potential can come down there, it can cause these vesicles to go and dump their contents into the synapse and release that neurotransmitter, which can talk to the next cell or send a signal to the next cell. There is a lot of cytoskeleton involved in this, a lot of microtubules, microfilaments, intermediate filaments. Altogether, we kind of call these things a lot of neurofilaments and neurofibrils. They provide that strength to that neuron so it can stretch a little bit when you're moving your limbs and things like that. And we are a lot of times shipping a number of different things up and down these axons. And in some cases, these axons can be a meter long. So if you think about a motor neuron leaving the base of the spinal cord and going down to the foot of somebody that's 6 foot tall, they might have a 3 foot long leg, like 6 foot, 6'5", 10, let's say, it can be a meter long axon that's going down there. So we are a lot of times going to be sending a lot of different things up and down this axon. If we're moving stuff from the cell body to the synaptic knobs, that is anterograde. If we're moving stuff back up from the end of the axon back, that is retrograde. And a lot of times we're going to be sending new materials up or bringing back broken down materials. So we actively use ATP to do what is called fast axonal transport. So 400 millimeters a day. So 10 millimeters is a centimeter. So we're talking 40 centimeters a day. This is using the microtubules and ATP. This can be anterograde or retrograde. So we're moving a lot of vesicles and other things down to the end there. Used vesicles, other harmful things we might be moving back up to the cell body. Axon, the cytoplasm within the axon, the axoplasm can flow. That travels at a much slower rate, but that is how we are going to do some of this slow axonal transport. So things like enzymes, new cytoplasm for the axon, that is how a lot of that is going to move down there. So again, much slower towards that end. And again, it's only slow axonal transport is only in that anterograde. The other thing that I want to mention about here is we do have different types of neurons. We've talked a little bit about the one type there with multiple dendrites, one cell body, and then a single axon leaving. That is what we refer to as a multipolar neuron. Many dendrites, single axon. There is also these things called bipolar neurons. You actually see them in the retina quite a bit. They have the cell body in two main processes, one main dendrite coming in, one main axon leaving. There's also these ones on sensory neurons that are called unipolar neurons or pseudo-unipolar neurons. These have an axon that really goes from the one end past the cell body that is kind of off on a little branch to the side like a T. And it's going to take that, like I said, the sensory neurons is taking that information into the central nervous system. And I believe I have a picture of each of these. There's also not many of these ones, but there is local anaxonic ones that do not have an axon. They have no dendrites or no axons. Again, these are not going to produce action potentials, just doing some minor local stuff in certain areas, but these are not going to be sending messages from one area to another with these ones. So this shows you that example. So multipolar neurons are mainly what we're going to be talking about, a lot like a hand. Multiple dendrites, one main axon leaving. Like I said, a bipolar neuron, it has branching on each ends here, so collaterals here and some collaterals on the main dendrite here. But you can see one main dendrite taking information in, one main axon leaving. We see these in the retina quite a bit. This is that unipolar neuron, like we said with a little T. You have one main axon that goes from the dendrites right down to the collaterals here, where the cell body is not being involved in any of the processing. It's just sending this down here. We see these a lot on sensory neurons that are just relaying stuff from the stimulus, where the receptor is, to the central nervous system at another synapse here. And then, like I said, those anaxonic ones here, these do not have any impulse conducting things here, just local action, local changes to the electrical activity on these ones. In reality, we're not going to talk much about these. These are also things that I want you to think about when we start talking about how action potentials travel. We're going to talk a lot about the dendrites and graded potentials and action potentials in the axon hillock. Realize we generally talk about on a multipolar neuron, so all the processing is going to happen here and then the signal gets sent here. It's going to be a little bit different on a neuron that's shaped like this or on a neuron that's shaped like this. We're going to see a little bit of, in terms of difference, where there may be an action potential from here to here, processing again, and then an action potential here, where this one, we're going to generate an action potential here that travels all the way down to the end and doesn't pause at all at the cell body. So keep these in mind as we talk about this a little bit, how the information is going to travel through a cell might change a little bit if we had a slightly different structure than the standard multipolar neuron at the top there. Again, how we can also classify neurons is based on what they're doing. So we can talk about sensory neurons. These are mostly, like I said, unipolar, a few bipolar neurons in here. So like with the sensory neurons, we have the cell bodies all in that what's called the dorsal root ganglion that we'll see in AMP2 outside the central nervous system. Motor neurons are almost all multipolar neurons. These are taking information from that central nervous system out an axon to those muscles or other effectors. And then again, on this one, most of the cell bodies on these motor neurons are in the central nervous system, and it's mainly just the axon that's going out. Interneurons are the other type that we need to worry about. These are nerves that are entirely within the central nervous system. These are the ones that are do a lot of these processing, that parallel processing we talk about, how we determine what we should do to respond to something. That is the vast majority of neurons. And again, most of these are also multipolar. But 99% of the neurons in the body are actually these interneurons. There's very few relative to the amount of neurons, very few sensory and motor neurons. And this can kind of show that example. You can have a sensory neuron close to a receptor here, takes its information back. You can see its cell body is in that dorsal root ganglion. It goes back. Here's an interneuron here. It's going to synapse with that one. If this was just a spinal reflex, it's going to synapse with that and send a message back out a motor neuron to, let's say, the skeletal muscle here. Or this could go to another neuron here that could send information up to the brain, and we could do the processing in the brain. So these different processes, a multipolar neuron has many dendrites, single axon, bipolar neuron, one dendrite, one axon, unipolar, shaped like a T. It's really just kind of a continuous axon, and anaxonic would have no axon. It's all dendrites. Another thing to kind of talk about here is how we term a lot of things here. So when we start talking about nerves in the peripheral nervous system, we call a bundle of these fibers a nerve. And these nerves are going to be surrounded with different connective tissue. So overall, we're going to have, and if we were actually doing dissection this semester, you would see that the nerves are actually quite large. It looks like wiring in the body of the cat. So you have an overall connective tissue membrane that is surrounding its dense irregular connective tissue surrounding the whole nerve called the epineurium, very much like the the epimysium. We then are going to have little bundles of axons together called fascicles, perineurium, like it was a paramecium in the muscle, and then each axon is going to have loose connective tissue, that aerial connective tissue surrounding it, called the endoneurium, very much like we talked about with the endomysium. And you can kind of see that here. These fascicles right here, perineurium surrounding that, epineurium surrounding the whole thing here, and each little axon here is going to have an endoneurium surrounding it. And this would be in the peripheral nervous system. We call this a nerve. Outside of that, we can talk about where they're coming from as how we classify some of these different nerves. So cranial nerves are going to be the ones that come out from the brain. We have 12 pairs that we'll talk about at some point in AMP2. The spinal nerves, we name these based on where they're leaving the vertebrae. They're kind of numbered. The first spinal nerve is going to be with the cervical vertebrae, so we talk about it being the C1, C2 nerve, T1, T2 nerve, all the way down to the bottom. And again, 31 pairs of spinal nerves coming out from that spinal cord. So you can kind of see the cranial nerves as these little stumps leaving off the brain stem, as well as right here, and the optic nerves right there. You can see the spinal nerves branching off the spinal cord at these different areas up and down through that. Again, sensory nerves functionally are only sensory neurons. Motor nerves are there. We're going to see a lot of these spinal nerves as well as cranial nerves are going to be what we would term as a mixed nerve that is going to have axons of both sensory and motor nerves bundled together that are traveling in this nerve. For example, the optic nerve is a sensory-only nerve. The oculomotor is a motor-only nerve, and then you have things like the trigeminal, which is both in terms of some of these different functional classifications of those were examples of cranial nerves. Pretty much all of our spinal nerves are mixed nerves. So that kind of talks to you a little bit about the functional and kind of structural classifications of the different nerves. What we're going to start getting into now is a little bit more of the physiology of this and how these nerve impulses are going to happen, how they're being triggered, and how they're processed. So I will see you again next time.